Fuzz, today we are going to talk about DNA wires. So, as an electronics engineer, a wire is typically made of a conductor, um, typically, and so it's a kind of a metal, and you will have electrons that will flow in the metal structure, because metal alloys have this function of giving freedom to electrons to pass through them when there is electric potential. So a current happens. So this is what metals do. And of course, we can also use semiconductors for that with some twist. But DNA wires seems like quite peculiar, because like DNA, the way you have explained it is, those organic materials hooked up together. So I always thought that DNA was mainly for information storage. What does it mean to have a DNA wire?
Yeah, well, I've heard some people say that DNA is a really strange molecule. And I don't know if I would say that it's a strange molecule, but it sure has some remarkable properties that are really go outside of what we would typically think about when we think about DNA. This might be a topic for a future episode, but one use of DNA is to create essentially nanostructures. This is called DNA origami, where you can literally create these very complex three-dimensional structures using DNA, kind of like a scaffolding.
Wow.
And so here people are thinking about DNA more in a material science standpoint than really thinking about DNA as a biomolecule that's storing information, that's storing the blueprint of life. But another really fascinating property of DNA is that it can function like a wire. And maybe we could just put up one of the slides real quick.
Yeah.
Just to give people a picture. This is a picture of DNA. And by the way, you and I recorded a short segment about 20 minutes long about the structure of DNA. So we're not going to review that here, but we can direct people to that particular recording if they want to get a little bit of background information on DNA. But
sure.
Here we see DNA as essentially being a molecule that consists of two molecular strands built from subunit molecules, where the sequence of those subunits, the adenine, guanine, cytosine, and thymine, sometimes called the genetic letters, is where the information is harbored. And of course, those two strands twist into a DNA double helix. And oftentimes, when we think about DNA, we think about it like we see in the bottom picture there of DNA, which is a space-filling model, where we see the positions of all the atoms in DNA. But if we think about DNA, or if we view DNA in a different orientation as we look down the axis instead of along the axis, we begin to see something rather peculiar. And that is that in the interior of the DNA double helix, there are the nucleobases that are stacked on top of each other. Those nucleobases are planar molecules, and they have aromatic bonding, which means they have electron densities above and below the ring for each nucleobase. And because the nucleobases are stacked on top of each other in the interior of DNA, those electrons from each of the nucleobases will overlap. The electron clouds will overlap to form, in effect, a molecular orbital that goes through the entire length of the DNA molecule. And that molecular orbital now can serve, in effect, as a wire. And so in this image, we see, again, the DNA double helix. On the left, we're looking along the axis. On the right, we're looking down the axis, where, again, the yellow nucleobases that are depicted in yellow are seen to be essentially overlapping as we go down the length of the double helix. And here's another picture that shows, in effect, the what's called pi-pi stacking. So here we take a little piece of the double helix and expand the interior region where we see the different nucleobases. And you can see that there's these electron clouds that overlap with each other, and they, in a sense, form this what's called this pi-pi stacking interaction. But that pi-pi stacking interaction goes along the entire length of the double helix. Here's another diagram showing, again, that same depiction in different colors, just so, again, people can appreciate, again, what's happening in the interior of the DNA molecule. Well, it turns out...
Actually, when you look to it from this top view, this cross section, it already looks like a wire with an insulator around it. Just gives the impression.
Well, there's actually something to that statement. That's really very insightful, Ahmed, because the nucleobases are actually water insoluble. They're hydrophobic. They want to avoid water. And the backbone of DNA, which is made up of an alternating sugar phosphate, alternating sugar phosphates along the length, are hydrophilic. They are water soluble, where the phosphate group actually bears a negative charge. And so you have with the DNA double helix, the interior is hydrophobic, the exterior is actually hydrophilic, and that difference, because DNA exists in water, that difference actually helps to stabilize the structure of DNA. So it's almost like a surfactant, if you will, where part of the molecule is water insoluble, part of it is water soluble. And that actually forces the DNA to form this double helical structure in order to satisfy both portions of the molecule. And what's interesting is the hydrogen bonding interactions that occur between the strands that also help stabilize the structure are not something that you would necessarily think would provide stability in an aqueous environment. But the reason why that molecular recognition works in the DNA double helix is because you've created this hydrophobic environment. So it very much is like an insulator surrounding a conductor, if you will. There's a, that's a really insightful, comment from your part.
So if I just try to imagine this in 3D with time, if we have, let's assume we have this ladder, and it is not, it's unwound. We unwind it, and then it's not in water. But once we put it in water, because the inside wants to be really inside and the outside doesn't care, it's as if the interaction with water will cause it to be winded or wound about its axis to the extent that the exterior will wrap around the interior. So essentially, the interior of the double helix will be as hidden as possible from the water. Is that correct?
That's exactly right. And when you do look at the structure of DNA, you do see these grooves.
Yeah,
they're called major and minor grooves. And so some of the functional groups from the nucleobases are in those grooves, but those functional groups are ones that are going to be hydrophilic. And so so it even with the nucleobases that are exposed, the portions that are exposed are actually portions that would favorably interact with water. So it's really very much a protected environment in the interior of DNA.
Wow. And that's why when there is something about reading or replicating, then there is those machines that come and open it up, and then there is this bubble there is a bubble where it's like forcibly open, and then it can be read. Is that actually why it does that? Because otherwise it's completely inaccessible.
That's right. Yes, that's exactly right.
Wow, amazing. So the more we look into this, the more the marvels of how God has created that really unravels with unraveling the helix.
Well, back to the idea that the DNA wires, the person who I think probably played the most important role in discovering this was a scientist by the name of Jacqueline Barton. And in the 1990s, she discovered that, again, DNA has these wire properties. And it's very interesting because you were mentioning earlier about conductors, right? You typically
Mm-hmm.
think of conductors as being, metals, but there are some organic compounds that are conductors, like graphite, right?
Yeah.
When I was, a younger man, I played some tennis, and it was, in those days, it was a big deal to get a graphite tennis racket, right? It had better flexibility and whatnot, but you never wanted to play uh, tennis in a thunderstorm with a graphite racket.
No, you you will get electrified.
Right, and the and graphite, of course, consists of these fused carbon rings that are planar, and then you have these sheets of graphite that stack on top of each other, and you get the same kind of pi-pi interactions, and that's how current is conducted through graphite. It's because of
Wow.
that pi-pi stacking for the that takes place among the graphite sheets. And so Jacqueline Barton thought, well, that's similar to the interior structure of DNA. Could it be that DNA could actually conduct electrical current? And so she did a series of some incredibly wonderful experiments um, looking at the wire properties of DNA. And so the experiment that Jacqueline Barton did to demonstrate the wire properties of DNA is really an elegant experiment where she synthesized in the lab a short piece of DNA and attached or tethered to each of the ends of the DNA molecule two different inorganic compounds that had metal centers. And one of those compounds was a photoreducer, and the other was an oxidizer. And so when she shined light on the system, that photoreducer gave up an electron, which passed through the interior of the double helix to the DNA wire, and essentially uh, reduced the compound at the other end that existed in an oxidized form. And so in this diagram, you see essentially the light interacting with the green inorganic compound that would be kicking out one of its electrons. It would travel along the length of the double helix, and again interact with the inorganic compound that's depicted at the other end in red, which would pick up that electron. And they were able to measure the flow of the current through the DNA double helix by essentially using the red inorganic compound, the inorganic compound that was reduced, as kind of a probe or a reporter molecule or a reporter group that told them that again electrons flowed along the length of the DNA double helix. And there's a whole set of experiments that Jacqueline Barton in her lab did to further confirm the DNA wire properties. One of the things they discovered that is really interesting is that the flow of the electrons through the wire is actually appears to be through a quantum tunneling mechanism. So it's a very
Wow.
rapid movement of electrons through the double helix. And on top of that, they also showed that if there's any distortion in the double helix whatsoever, let's say due to a base pair mismatch or damage to the DNA, that will actually cause the wire to be disrupted. It
Wow.
breaks the wire, and so this becomes a very powerful sensor that can be used to characterize or to monitor different biological phenomena.
Wow. So let me get that right. So number one, the experiment was we will throw a ball from one side, and since the ball arrived at the other side, it must have passed in between. So this confirms that DNA essentially passes electrons, balls in this parable, and so like a pipe. So if you throw one electron from one side, you can pick it up from the other one. And the second thing is that this only happens if DNA is in its intended structure. So if it's not exactly the way it should be, it just doesn't do it. Is that correct?
That's exactly right.
Wow.
And so this becomes again a very powerful way to ensure the integrity of the DNA molecule. And apparently this wire phenomena is hypersensitive to any kind of distortion in the DNA double helix.
Wow. So now, by the way, this reminds me of a very important problem in electrical current distribution. Okay, because we have those huge electrical current distribution networks and the overhead cables and everything, and then guess what? Suddenly, a part of the city loses electricity, and then you need to know where exactly the wire was cut. But I will wait. I will wait until you tell me what are the applications of this in the cell, and then let's see how God has solved the problem, and how we were solving the problem.
Yeah. Well, not to get too ahead of ourselves, but it, you know, that is a real challenge to figure out where the wire is broken, right? Where the
Yeah.
wire is cut. It's not not a trivial thing to do, and we'll see a really elegant solution to that problem in DNA in just a minute. But something else that Jacqueline Barton and her team did as well is to also do other experiments. For example, where they would essentially tether the DNA double helix to a gold surface. And what they discovered is that this was an excellent way to measure the binding of proteins to the DNA that have what's called an iron sulfur center.
Hmm.
And this is a very common structure that you see in biochemistry, is kind of an iron sulfur complex that is essentially a cofactor with enzymes. And it turns out that a lot of the DNA binding proteins actually have this iron sulfur moiety. And these can exist in an oxidized and a reduced state. In the oxidized state, the iron is a +3. In the reduced state, the iron is in a +2 configuration. And so, for example, there are experiments that they did where they had, for example, a particular DNA binding protein that where the in the iron sulfur cluster would be in a reduced state, in the +2 state. And if it bound the molecule ATP, that binding would actually then cause that protein to bind to the DNA. So it was an ATP mediated binding of that protein. And when it would bind to the DNA, because the DNA is tethered to a gold surface, that would cause that iron in the +2 state to become oxidized, sending its electron along the length of the double helix to the gold surface. And if you reversed the electrical potential, you could actually cause the movement of electrons in the opposite direction, so that electrons can move from the gold surface through the double helix and essentially reduce the iron in that iron sulfur cluster. And so this is another neat experiment that demonstrates, one, the wire properties of DNA, but it also demonstrates the fact that that DNA can serve as a probe to measure protein binding, and that when proteins bind to the DNA, if they have, again, metal centers like an iron sulfur cluster, they can actually send electrons along the length of the double helix. And this actually points us to a very important biochemical application of this DNA wire phenomena that takes place inside the cell, that allows the cell to survey for DNA damage.
Survey for DNA damage. Okay. Okay, that's interesting.
So let's go ahead and show you how that works.
Yeah.
And so
So
this is
DNA damage is something that happens every day and every minute, right? DNA is getting damaged because of so many reasons, right?
yes, yeah. So it goes back to the idea that one of the primary biological roles of DNA is data storage. And so the integrity of that data has to be maintained. And DNA is in an environment in the cell where it is exposed to chemical insults, UV radiation impinging upon the cell will cause damage to DNA, chemical insults. There can be other processes in the cell that cause base pairing mismatches. Let's say an error that takes place in DNA replication, causing a base pair mismatch that needs to be corrected. And the cell has a correction mechanism. It's called excision repair, where the region of the DNA that's damaged or that is mismatched will be cut out. And then there are enzymes that will come in and essentially replace the damaged DNA with DNA with subunits that have chemical integrity, and then that's all sealed back together, and so that repair is affected. It's kind of like the repair you would do on a wire that would be damaged, right? Where you would come in and cut out the broken part and splice together a new wire, creating in that
Yeah.
power line, let's say, that was damaged, right?
So one thing is that damage is happening all the time, and damage is, this is plenty of damage, like thousands or millions of damage incidents a day, right?
Yeah. Oh, yes. Yeah. This is happening all the time, and that damage needs to again be repaired, because if it isn't, you're losing again the integrity of the information stored in the DNA, and that has some very serious consequences to the organism, because it in effect is a mutation in the DNA and a loss of that of critical information.
So
And
the
so,
second thing is that the system needs to know whether damage has happened and exactly where.
right,
Yeah.
and so when you think about DNA the amount of DNA in cells is enormous, right?
Right.
In human cells, it's 3.2 billion base pairs, of DNA that constitutes the genome, and it's distributed among 23 different chromosomes, but that's still a massive amount of DNA. And so, how do you survey that DNA to determine if it's functional or not? And it turns
And
out,
that's in each and every cell almost.
yes, that's right.
Yeah.
And so one way in which this appears to be done, at least in microorganisms, and presumably this is also happening in eukaryotic cells as well, is that you have these surveillance proteins that bind to DNA that have a iron sulfur cluster. And
Wow.
so, so there are two different proteins that are shown in this diagram.
Mm-hmm.
And when a protein binds to the DNA, it's going to be in its reduced form, and it'll send an electron along the length of DNA. And so, in its oxidized form, when the iron is in a +3 state, that protein will remain bound to the DNA molecule. And when another molecule that's bound picks up that electron, it can dissociate from the DNA, and then it'll reassociate with the DNA in another location. And when it does, it'll send an electron along the double helix, and that'll be picked up by, again, another protein in the oxidized state. And that process just goes on back and forth, where these proteins are just sending electrons back and forth between one another along the length of the DNA molecule. Now, if damage takes place, it disrupts the wire properties of DNA. And when that happens, when the wire properties are disrupted, now the electrical current can't flow. And so that DNA binding protein that is in that +3 state is now stuck to DNA, and it can actually very slowly move along the DNA double helix, but it's going to essentially be bound to the DNA in the location near the site of the damage. And over time, you're going to start building up, these molecules that, again, uh, um, near the site of the DNA damage. And these molecules actually have the excision repair capabilities. And so they will then slowly migrate to the site of the damage, replace that damage, the electrical current is reestablished, and they will become disassociated. And so it's a
Wow.
very elegant mechanism for very quickly and efficiently finding damage to the DNA and then repairing it, where you really need a very small number of the DNA repair proteins to actually affect the repair process because of this surveillance mechanism. So this is absolutely astounding to me, that not only is this really unusual property of DNA actually useful for nanotechnology, but it actually is a mechanism that is integral to DNA. And what's intriguing is the fact that many DNA binding proteins, again, have these iron sulfur clusters in them,
Wow.
suggesting that this wire mechanism may be used for uh, other processes that involve DNA. It may not just simply be limited exclusively to DNA repair. And so this is
Wow.
something that Jacqueline Barton's group is continuing to work on, is really trying to establish the biological extent of this type of mechanism being used inside cells.
Wow. So let me make sure I got that right. So there are those couples of proteins that have the iron cores, and if it goes to, if it binds to the DNA, it releases an electron that goes to another one that's already bound and causes it to release if the connection between them is intact. So if the whole DNA was intact, they will just keep, one jumps in, it causes another one to jump out. So they would just, you know, juggling and jogging and running all over the place until one of them lands in a place where there is discontinuity. So the wire is cut between those two guys in the third image. So they will be stuck there because the electron cannot jump between them. And since they are stuck there, it means that the damaged location is in between. And then the rest of the molecules are recruited, and then the repair mechanism will happen. So in this case, this motion, because when I used to watch those rendered graphics, they would show you a piece of the DNA where there is damage, and then there are those proteins parachuting, you know right to the damaged location, and then you need to think, how does those guys know that there is damage here? Are
Yeah.
they like parachuting at each, or do they get a radio signal or something? Come on, they're calling damage repair crew. There must be something to it.
Well, and this is how they do it, and you know, and this is really a fairly recent recognition, that this is how, again, that the proteins can very quickly find the DNA damage and repair it. So it's an incredibly ingenious mechanism, uh,
Wow.
and it just speaks to the fact that every aspect of the DNA molecule is serving a critical role in terms of its role as a data storage system, because this wire property is critical for DNA to be able to store over long term data with integrity.
Actually, this is really amazing. And you mentioned that there is few of those. So I'm not really few, but given that we have like 3.2 billion base pairs in the human genome, those are not in those would not be in the billions in the cell. There's much, much, much less, but they're jumping around all the time looking for it, right?
Yes, yes, and I'm not sure what is the length of DNA that these proteins would survey, right? But um, you could envision in several thousand base pairs, for example, between the proteins, and so it gives you the ability to very quickly scan vast amounts of DNA. Something that that's I think worth noting here is in bacteria, uh, DNA exists as a naked is as naked DNA. There's no real proteins permanently associated with it. In eukaryotic cells, cells with a nucleus, the DNA is actually wound around histone. It's associated with histones, and so you have a histone core where DNA winds around that core, and then there's other histones that bind to the linker regions of the DNA, and then that all kind of coalesces into, these dense structures that we can visually see called chromosomes. But one of the questions that Jacqueline Barton's lab asked is, can DNA current flow through DNA when it actually is wrapping around uh, these histone cores? Because even though the DNA structure, the double helix is linear, depending upon the sequence, you actually can introduce curvature in the helix. And that curvature is how it winds around the histone core. Well, it turns out that you can still have electrical current flowing through DNA when it winds around a histone core. So that overlapping is still, and the wire is still preserved, even though the helix may actually have curvature, but it gets disrupted if there's any kind of damage or any kind of base mismatch. So, remarkable sensitivity,
Yeah.
and remarkable
Remarkable
robustness.
sensitivity when there is damage, but when there are other things that are needed as part of the design, like the histone wrapping, it just does no harm. So completely fit for the purpose, right?
Yes, exactly. Well,
Yeah.
and to me, I think as we talk about this, what becomes evident is just, again, how remarkable, how marvelous, you know, uh, biochemical systems are. I mean, the terms I would use would be sophistication and elegance and ingenuity, and that, of course, is consistent with the idea that biology is the product of a creator's handiwork.
Yeah.
That this
Absolutely.
points to a creator. But one of the things that I did in a book that I wrote and published a few years ago now, called The Cell's Design, is I pointed out that there are these remarkable similarities between human designs that we invent and engineer and the structure and function of biochemical systems. And I use that to kind of present a modern day version of the watchmaker argument, that we, when we design things, objects or systems or devices, those things we make have certain properties that reflect the work of a mind, and that when we look at biochemical systems, we see those very same properties that suggest the work of a mind. But one of the things I did in the book is I advanced what I would call the watchmaker prediction, which is the idea that we see a creator's handiwork when we recognize similarity between our technology and the technology of found inside the cell.
Hmm.
But I make the prediction that as we invent new technology, we are going to see examples of that new technology inside inside the cell. And so that's the watchmaker prediction, that as we invent new technologies, we'll recognize, well, those technologies already are implemented in the cell's biochemical systems. And so this idea that DNA wires that were discovered by Jacqueline Barton in the 90s was considered to be a really interesting phenomena that was really something that fell under the umbrella of material science and nanotechnology. It was only years later, actually after The Cell's Design was published, that she asked the question, is this biologically relevant? Because people immediately saw the use of that uh, uh, insight for technology purposes. It was only later that she asked the question, is this something that happens in biology? And lo and behold, she discovered that that the cell is using this technology in much the same way that nanoscientists and material scientists would think about using the technology. So it was it satisfied the watchmaker prediction, which is kind of neat. In other words, it's saying that the case that we have for a creator through a watchmaker argument is actually much stronger than we even realize, because we just don't recognize the technology in the cell. because we haven't invented that technology yet to recognize it.
So what you're saying is that not only the technology in the cells at a point of time when we look at it, we find it similar to what we do outside, confirms that it's the product of a mind, but also that there are technologies in the cell that when we will see and come to understand, it will teach us to advance our technology better. Because would you say that this is because this is a natural prediction, if we take the hypothesis that this is designed by a supreme wisdom, and hence the technologies there must be so advanced to the extent that when we understand it, we will essentially get to use it and advance ourselves?
Yes, that's exactly how I would think about that. That that if indeed biology is the product of a mind, then we would expect that biology should be a source of inspiration for new technology, for engineering. And in fact, there are areas of engineering today called biomimetics and bioinspiration where engineers just
Biomimetics.
they just
Yeah.
copy the designs in biology. And a lot of times these by copying these designs, there are engineering problems that are solved that apart from that are very challenging, maybe intractable or apparently intractable problems. And so to me, if biology is the product of an unguided, historically contingent process, you would never expect it to be, number one, riddled with these have these elegant designs pervading biology, but it wouldn't never be a useful source of insight for technology and for engineering.
Yeah, absolutely, absolutely. You can only learn from something that's more advanced. Absolutely. And I'll tell you the analogy with electrical engineering. So I have those huge networks of wires, and in the older days, somebody had to go and survey the wires. So he would have to go over the length and some 10 kilometer cable, and then you'll need to go through the 10 kilometer. But lately there are those modules. Not so lately, but they got better, and you really attach the modules along the power lines to the extent if a break happens, then the two modules around the break would be the first to know, and then you get them connected. Maybe if you're using an advanced technology, to the cellular network, and then or they can connect to each other by radio, and then one of them will be a central for communication with the GSM network, etc. But the funny thing is that we really put so many of those modules all over the place. Now, the ingeniousness of what you have described is that the genetic system is really using fewer of them, but they're jumping around all the time. It's unbelievable. So otherwise, you would have those 3 billion, and if it's like 1 to 1,000, you need 3 million of them. But it looks like that it's really maybe less because they are really going all over the place all the time until they find the faulty place, and then they start recruiting. And I would really expect that this recruitment process has something into it more than molecules accumulating around somebody that's stuck somewhere, because they really look pretty intricate. I cannot imagine that they're recruiting just by chance. What's your thoughts about that?
Yeah, well, I guess I haven't given it much thought. How's that for
Yeah.
in terms of how this is actually happening? The model that Jacqueline Barton presented is that the enzymes that are doing the excision repair are actually the enzymes that are also doing the surveillance for the DNA damage. And so once they are oxidized and they can't pick up an electron because the wire is broken, they then are going to just slowly move along the DNA until they find that site of damage. So it would be, the analogy would be, you have the modules, that, help to pinpoint the location of the line break. But if those modules, when the line broke, were able to then slowly migrate till they found that the point of the break and then somehow magically repair it, right? That's in effect what's going on. So there may be some recruitment that's taking place for the repair enzymes, but the repair capabilities are actually in the proteins themselves. So it's a really, again, a really elegant system.
Absolutely. And you also mentioned that this is useful for the replication, not only the damage repair.
Yes, and that's something that's we may want to pick that up when we talk about DNA replication at a future point in time. But essentially, the when a DNA strand is replicated, so you have the DNA double helix that conceptually separates, and this is happening at a replication origin site, the replication will occur along both of the strands of DNA, but there's a directionality to the replication. But the first thing that happens is a large complex called a primosome lays down a piece of RNA, an RNA primer, and then once that RNA primer gets to a certain length, a DNA polymerase will bind and build off that RNA primer to begin the process of DNA replication proper. And it's during that transition where the primosome kind of dissociates from the DNA and the DNA polymerase binds and begins its process that wiring mechanism is working as well, where there's again that because the enzymes involved are iron sulfur cluster enzymes, and so the thought is that by establishing and then breaking electrical current, that actually is signaling both of those enzymes when to stop and when to start respectively. So it's it's a bit of a complicated mechanism that I just figured it's probably this is enough to make the point. But again, it's the idea that this wire property is being used for the different enzymes that are working on DNA to signal one another and to control what they're doing by passing electrical signals back and forth or electrons back and forth between each other.
Fuz, what would you say? The way I get it is DNA is, it has this core, it's connecting or it's passing electrons through, and the rest of the system is exploiting that feature to the extent that the proteins and the enzymes that are responsible for certain functions that need signaling actually have metal cores that would send or receive electrons. So it is like we have transmitters and receivers and the wires. So it's an integrated system.
Yeah.
What would you say to somebody who would tell you, well, this is all there by chance? Is that even a possibility?
Well, you're looking at what Michael Behe would call an irreducibly complex system, or maybe you could call it an integratively complex system, right?
Yeah.
Where the whole system has to be fully intact for it to be of any value whatsoever. And so
and in this example in specific, we know that if there is anything that harms this intactness, that it really doesn't function. But the amazing thing is that it will look for the damage to fix it because it wants to be intact.
Yes. So it's very hard to envision how a system like this would come together in a piecemeal manner. It seems like it has to be instantiated all at once in order for that system to have any value whatsoever for the cell. And so it's it's again, I think an indication that this looks like it's designed. It looks like it's the work of a mind.
I have another insight I want to take your opinion about. If the system of the cell is going through so much trouble to find damage, to fix. This damage, if not fixed, that would constitute what we call a mutation, right?
Mm-hmm.
It's going through all that kind of trouble to fix this damage. But we have another theory that claims that every advancement of life is because of damage. So I'm wondering, does it make any sense that the system is fighting its own advancement, or rather that no, it is not mutation that's driving anything? Mutations are identified by the system as harmful, and they are all the time being fixed, and cutting wires will not convert a TV into a computer. And the
Yeah.
TV knows that. It wants to remain a TV. It doesn't want to degrade, essentially.
Yeah. Well, I mean, what you've identified there is, is the paradox, right? Is that, if evolution is being driven by mutational events, changes in the genetic material, then you would think that having a repair mechanism in place would actually be counterproductive to that. But on the other hand, if you have mutations running amok, organisms are not going to survive long enough to actually reproduce. So it really represents a paradox of sorts. And I mean, one way that maybe an evolutionary biologist would respond to that would be that if you had a repair process that was operating at 100% efficiency, that would be so expensive to the cell that it would natural selection would prevent that 100% repair from taking place. So they would argue that the repair takes place as a trade-off where enough of the damage is repaired to ensure the integrity of the organism, but because not all of the damage is repaired, it allows for some mutation. So you would have to argue that it was a fine-tuned process to even allow some mutation. So I would suspect that's how an evolutionary biologist would respond to that. But I think your point is really, a solid point, is that you do have a paradox, and that you have these competing processes.
Yeah. Because we know that by our experience, when somebody causes the mutation rate in his system to be beyond what the system can fix, for example, by smoking or drinking alcohol or whatever, which causes chemically induced mutations, that he gets sick or contracts cancer, or what have you. So essentially, we have never seen a human turn into an X-Man by smoking too much. Or getting drunk all the time. We have never seen a real Hulk generated by being exposed to gamma radiation. It only happens in Marvel.
Yeah.
not any other place. So it seems that this system knows what it's fixing, and the message there is, when you have instructions to properly behave in life and respect your body, the system just works fine. If you push it too hard, you're just harming yourself. And if it's something natural, then the animal that gets diseased is eaten by the lion. The slowest gazelle will be eaten by the lion. Natural selection will just cause the mutated gazelle to be eaten.
Yeah.
So I really don't know, even if you argue for some few mutations, what are you going to do with the lion?
Yeah.
That's how I see it. So I think this was a great session, Fuz, and really, I hope everybody has enjoyed it. And guys, what you have put in the live comments, we will be responding to in the after show. So make sure to jump there, and you will find the link to it in the live stream. And Fuz, thank you very much. And I'm really looking forward to the upcoming episode. Guys, just be sure to go there and click the notification bell for the next episode, which will be about parity code